everyone, my name is Daryshenee LETCHUMANAN and my metric number is 202349. Today I'm going to demonstrate the procedure for streaking single isolated colonies. Please make sure you wear your glove and also lab coat before you start your experiment. And I have already sterilized this table. Before we start the experiment, let's look at the principle for streaking single isolated colonies. The streak plate method is a rapid qualitative isolation method. The techniques commonly used for isolation of discrete colonies initially require that the number of organisms in the inoculums be reduced. It is essentially a dilution technique that involves spreading a loopful of culture over the surface of an agar plate. In the streaking procedure, a sterile loop or swab is used to obtain an uncontaminated microbial culture. The process is called picking colonies when it is done from an agar plate with isolated colonies and is transferred to a new agar plate using a sterile loop or needle. The streaking process will dilute out the sample that was placed in the initial region of the agar surface. Pure colonies an individual colony that is transferred to another plate to produce pure culture or consisting of one type of bacteria. These are the materials and apparatus needed for this experiment. First, divide the plate into 4 quadrants. Flame a wire of inoculation loop until at least half the length is red hot. Next, allow the loop to cool, holding it near the flame but not in it. Sterile the opening and insert sterile loop to the center of a well-isolated single colony. Sterile the tube and close. After that, make the primary inoculation in the first quadrant of the plate. Keep the plate covered to avoid contamination. Reflame and cool the loop. Rotate the plate a 1/4 turn. Using the sterile loop, make a second set of 4 streaks that overlap a small area of the primary inoculum. Reflame and cool the loop. Rotate the plate a 1/4 turn again. Make a third set of streaks in the same manner as the second, just overlapping the previous set. Reflame and cool the loop. Rotate the plate a 1/4 turn again. For the fourth streak, start in the end of the third set and zigzag across the remaining area of the plate. Reflame the loop and return it to the rack. Remember to place the plate upside down so that the condensation doesn't drip onto the colonies and disturb them. Now that we completed our streak, we can place the agar plate in an incubator at 37 degrees Celsius for overnight or at least 12 hours. Now, let's look at the expected result of isolated mixed culture between S. marchensis and S. aureus. These are the description for the colonies. While this is the expected result for isolated mixed culture between E. coli and S. aureus. These are the description for the colony form. That's all from me. Thank you.